Hi, I'm Veron Ramsuran. I'm a professor in genetics at the University of KwaZulu-Natal in Durban, South Africa. In some areas in South Africa, we have HIV incidence rates as high as 60%, which is really scary. And my interest in genetics, which is what pushed me towards this area, because not much was done with regards to genetics in HIV in South Africa. So Africa has the largest genetics diversity in the world, and it's not being explored as much as we would like it to be. So this is where my research in genetics collided with my interest in HIV. So we started looking at what people called junk DNA, and actually this is not really junk DNA. This is pieces of DNA that we thought had no function before, which was hence called junk DNA. Now we realize that this DNA actually is responsible for several functions, including phenotypic effects and how people get infected with HIV and how long they progress with the virus. And the idea is looking at throughout the entire genome, how does epigenetics control the effect of HIV and how long people get infected for, how severe the virus is. And what we're looking at is we're looking at a mechanism called DNA methylation. DNA methylation is what we are planning looking at across the genome. So DNA methylation is when a methyl group attaches onto a DNA sequences and controls how a gene is being expressed. So when the methylation is present, a gene is turned off. And if that gene is responsible for protection against HIV, it actually is turned off, and then you get infected or you progress much faster. So we're now trying to find out if we can control these DNA methylation molecules, and if we can control how people get infected or when they get infected, or how to protect from infection. The second set of projects that we are looking at is COVID. A lot of attention has now been focused onto COVID disease. Using my expertise in genetics, we want to now find out why is there such a huge difference between the way people get infected with COVID. You have individuals that are asymptomatic, you have individuals that are symptomatic, some individuals are severely symptomatic where they require to go to hospitalization, and then you have individuals that eventually die from COVID. So the question is, why is this such a difference across individuals? It's probably not due to the virus, because individuals get infected with a similar type of virus, whether it's the Delta variant or the Omicron variant. But it actually comes down to the human genes that they are present. And what my research has found is that there are some human genes that confer protection against COVID disease, where we found that different individuals of different ethnicities have more protection than other individuals simply because of the frequencies of mutations that are found across these individuals. And what we find is that if we can try and mimic what's happening in that actual person's body in terms of protection, we can design better drugs or better drug target sites that can lead to more protection. Simply by looking at some of the day-to-day tasks such as PCR, real-time PCR, sequencing, this helps me a lot with all of my activities where I'm looking at simple mutation detection, expression of genes, amplification of products, and so on. Applied Biosystems is one of my go-to platforms for a new type of research that I'm doing, or just basic research that I'm doing in the lab. The best thing about being a scientist to me is making the new discoveries, is trying to see if we can actually change healthcare in the future, is training the new students, is seeing the joy on their face when their first gel works, when their PCR assay is working. It's a joy of giving knowledge to a younger generation. So one of the biggest things that inspires my research is making discoveries that have a potential of changing healthcare in South Africa. So I feel that the research that I'm doing can have an impact on people's lives.